So first of all, I thought I would start out this week. First of all, I have to say, Dr. Follett, that was an amazing talk. That's an incredible story. I just want to acknowledge what what he has accomplished. Um, Second thing is, to start this week, I wanted to start by talking about cancer. And in particular, what my lab focuses on is what I call the biology of therapeutics. So why is it that we've been at cancer for decades now, and yet the success rate in cancer clinical trials is still very low, as I'll show you, like very low. It's one of the worst. And while we've made great progress in cancer survival, we are far away from curing it, right? To the point where we're not even allowed to say the C word, can't cure anymore. And that's not right. All right, so let's get into all of this. And I'll show you some of the efforts that my lab is doing, including this guy. Actually, my second talk in Wednesday, I'll talk more about work that Hamid's doing, which I think is pretty amazing. But I'll show you how we're getting at some of the questions using flies, and why is it when I give you a cancer drug, it either won't work or it'll work for a little while and then stop working, and what we're trying to do about it. Okay, so we're into this. Groovy. Let's start with the fact that cancer is a fundamental, and as far as our communities are concerned, an ancient disease, right? So, for example, several thousand years ago, 1600 BC, there's a very famous medical treatise. It's known as the Edwin Smith Papyrus, but it's from Egypt, and it was written by a series of medical doctors there. It's actually quite a long treatise, and in that, they talk about 8 breast cancer patients that they treated. They treated it with cauterization, which means they try to burn it out. I want to remind you, this is before anesthesia, so that's no joke. And the other thing they note in there is that despite their efforts to burn it out, to cut it out, they still weren't able to cure these women, and they died of what we now know as metastatic disease. This was 3,500 years ago. We're a bit better, for sure. I don't want to dismiss that. We've great progress in breast tumors, and you had asked about flies and breast cancer, and we've done some work on this. But we can be a lot better, right? So let's talk about sort of the history of cancer treatments. So, and the most effective treatment that we have today in 2026 is surgery. And pretty straightforward. You have a tumor, you cut the tumor out. If you get all of it, you're good to go. And actually, 50% of cancer patients are just cured by surgery right off the bat. So it's by far the most effective still. Okay? But of course, excuse me, starting to go through puberty again. Of course, the little phrase that I threw in, if you get all of it, is the kicker. Because there's something called metastasis. I think you sort of know what metastasis is. Basically, if you remove the mothership, but some of the cells have left and gone elsewhere in the body, they're hard to track. You can't really cut them out, and they're a problem. So they begin to grow, you form secondary tumors, and over 90% of all cancer deaths are due to metastasis. That's actually the problem. So if I'm a woman, I'm not going to die of a tumor in my breast. That's not itself a problem. It's when those cells leave, get into my brain, my bone, my liver. That's the problem. Okay? And so if you want to get at cancer, you're really talking about cancer metastasis in most cases. Right? So let's get to the heart of that problem. And so through the 20th century, we got into secondary treatments, chemotherapy. So what is chemo? So I want to point out that chemotherapy is not the same as targeted drug therapy from a lingo standpoint. Chemotherapy has a very specific meaning, and that is usually metal-based, platinum-based drugs where the platinum ion just sticks into the DNA. And when you try to replicate that DNA, for example, you want to make a bunch of tumors, you'll damage that DNA when you try to replicate it, the cell will die. So anything that tries to divide your tumor, your immune cells, the stem cells for your hair, your skin, your gut, and so on, it'll kill all of that. And chemo, second line therapy, is, next to surgery, in 2026, the most effective treatment for cancer still. Okay? So we'll get to targeted therapies. It's what most of you know something about. They are not as effective as surgery or chemo today. Okay? So this was great. Radiation is another way of doing the same thing. You damage the DNA when you try to replicate it. The cell realizes, oh, I've made bad DNA. I self-killed. Somebody was talking about apoptosis. My lab has actually spent a fair bit of time studying apoptosis. So, and that is also very intimately related to cancer. So radiation, chemotherapy, that was pretty much standard of care through the 1990s. And I'm so old that I remember everything sort of there on. And that is that tumors eventually figure out a way to get around chemo, usually through transporters. They'll kick the drug back out of the cell. There's other ways to do it, but basically they find ways around it. There's a little Darwinian competition always going on there. You've got typically billions, occasionally trillions of cells going there. One of them is going to happen to be configured to get around what you're treating. Okay? This is annoying thing about cancer, all right? No matter what theory you have, if you get a billion of them, but one of them gets away, you lose. So it's tricky. And I have to admit, side note, that I actually was trained as a developmental biologist. I worked on the Drosophila eye and used it as a model for how you construct epithelia and stuff like that, and nervous system. And when we stepped into cancer, and I'll show you a story, I think in this talk, I thought, these cancer biologists, they must be idiots. They're not getting anywhere. This is so easy, right? So I worked out the hard stuff, which is how you develop the nervous system. How hard could this thing be? So I have to admit, it's actually a little trickier than I expected, and it's going to take us at least a few more weeks for this guy to figure it out. Low pressure there, Hamid. Yeah. And that is, in the 90s, when I started to get interested in this, there came this idea of precision medicine. And the idea is basically see target, hit target, which is I learn what I can about the tumors, really mostly about the mutations that are driving it. So for the most part, cancer is fundamentally a disease of DNA mutations. Okay? It's a little more complicated than that, but basically. And so the idea is you find the primary driver, you make a drug that hits that, and you target that in to choke the tumor out. And let's talk about how that's gone. And it's a little ironic that I'm the Regis Professor of Precision Medicine, because I've been sort of a nasty critic of this general approach. And that is because cancer is a very complicated network. This is where the brilliance of flies come in, as we'll get into. And the idea that you would pick a target, take it out, and the network would just go away and become normal is not real world. When you take the node out of a network, the network collapses. That's sort of the point. And that another word for that is, anybody interested? And another word for collapsing a network is toxicity. It turns out that precision medicine as a class is more toxic than chemotherapy. And that's hard to do. Some of you may know people who have taken chemo. That's no joke. But targeted therapies as a class have been really, really toxic because you're collapsing networks of proteins that they're so fundamental to the biology of your body that when you mess with them, one amino acid in them, you can get an entire world, entire growth, a tumor in the body. So rarely do side proteins cause tumors. It's always fundamental stuff. Okay? So these are the challenges that we have, and I'll get into some of the issues with that. Yes?
Now, this issue of precision medicine.
Yeah.
Well, I'll go into drug delivery. Probably using nanoparticle to deliver the drug on the tumor cell, bringing about limited toxicity to the major protein that are crucial to the body system.
Absolutely. It's a great question. So the question is, if I can repeat it, the question is, what about more targeted delivery of the drug right to the tumor? So there's two ways to do this. One is to physically deliver it, and that is done where you inject the drugs into the tumor, usually with a virus, so it can spread locally. The problem with that is the metastasis problem, which is you're going after the mothership. And finding the other tumors is a little bit like whack-a-mole. So you can inject it into a second one in the liver, maybe a third one, maybe it gets into the bone. But I think I'll show you scans today. A typical metastatic tumor is spread through the body, so that's tough. So the second way to go at your idea, which is an excellent one, I should say, many companies are devoted to this. The second challenge, though, is, so the idea is, well, then let's find something that is specific to the tumor and get the drug to dock with it, maybe package it into a nanoparticle, something like this, right? And there's a huge amount of effort on this as well. The problem that has faced, the challenge that has faced, is, first of all, finding something that is both specific and on the surface, right? If it's specific and internal, that's not helpful because you can't dock internally. And the second bigger problem is you put this thing out on the surface, so the problem is I'm a tumor cell. I put this weird protein out on my surface. Maybe it's a fusion, something like that. You drug it out. You kill me. But remember, I've got a billion cells going. Some of them won't put that out on their surface. And now what you've done, and this is, I'm sure many of you have seen where you get treated, the tumor goes away, and then it comes roaring back, right? And if you do the math, overall survival doesn't change, basically, or it's within a few weeks. The reason for that is you have hyper-aggressive tumors that are actually weirdly kept at bay by the more steady, like the turtle versus the hare. The turtles tend to dominate. You kill off the turtles, which are the major part. You open the niche to the hare, and they grows like bonkers. And so when it comes back, it comes roaring back, and overall survival doesn't change very much. So the real answer to your question is that is a promising approach. I don't mean to dumb it. I'm just pointing out so far why we don't do that regularly. We haven't figured out a way to get at those rare ones that you spend months working something up and then it just comes back. So these are the challenges. So yeah, to this. We're cool? We're good? Okay. So as I mentioned, my lab studies the biology of therapeutics. I'm going to talk about cancer this week. We also do rare genetic diseases. We're getting more into those. Slides are fabulous for this, in part because rare genetic diseases by definition are rare. And drug companies generally don't have an interest in going after them because the market's not big enough for all the effort that's required. And so there's a number of challenges that we work on beyond basic research. But one of the challenges you can deal with is flies are quick, they're cheap, and so on. And so, for example, let me just mention an advantage that maybe you haven't thought about, but we do think about quite a bit. So, for example, we have one project we're working on something called RASopathies, which is it's an inherited genetic disease where you have mutations that activate the RAS pathway. Okay, and you've got all sorts of problems. You're born with all sorts of defects. It gets worse. Eventually, your heart thickens and you die, and it's pretty nasty, actually. The problem is it's quite rare, so there's not a lot of interest from drug companies. But there's another problem, which is, so we have a lead drug, and we have a paper that's hopefully going to be in press shortly for this, and we're hoping to run clinical trials. And my partner over in New York, Bruce Gelb, is going to probably run this. But the challenge you have with these rare diseases is to run a clinical trial on 3 kids is not a thing, okay? So if you just took 3 kids and gave them 6 months, 2 of them would get better out of statistical noise. So giving them a drug is not helpful, okay? You need at least 20 people and a drug that really works quite well in order to get enough data to say this is actually helping the child. So let's continue. And especially when you're treating children, you don't want to give them something that's going to mess up their development and is not even effective on their disease. So the bar is reasonably high. So what we do when we develop treatments for rare diseases, we don't make one example of it. We make, in the case of RASopathies, we have now made 31 different flies that represent 31 different mutations that drive that disease. And we looked for one thing to treat the whole set so that when we recruit in the clinical trials, we can take everybody in that basket. So instead of 3 kids, maybe we can have 20, 30, 40 kids that we can actually test. Does that make sense? So these are the sorts of things when you have flies that you can consider. To make 31 mice and then grow them up and do a screen for a drug, that's not even a thing. Okay, the cost of that would be wild. Okay, so when you're working with flies, you can imagine more because more is doable. That's my point. In cancer, this gentleman works in something where we have 70 fly avatars for KRAS-driven colorectal cancer, and you typically screen at least 10 when he's doing drug development. Again, 10 different mice with different. First of all, these flies have between 6 and 16 different mutations in them. Do that in a mouse, and then to do 10 variations or 20 variations in a drug screen. Again, with flies, it allows you to imagine things that you just can't imagine doing in other systems. Okay? All right. So to summarize, in the lab, we build what are called fly avatars, and I'll explain what they are. But basically, each fly line represents a patient, right? And we've developed, especially Peter Smybert, postdoc when he was back in New York, developed technology to quickly create a 10-hit, 15-hit fly that mirrors the mutations in the patient. Okay? And we are in a clinical trials of. Now, what the lab is getting deeper into is pairing these with what we call human assembloids, just basically human in a dish. And that is taking from the local hospital, the Queen Elizabeth University Hospital, taking primary tissue through our collaborator, Joanne Edwards. So we have primary tumor tissue taken right from the patient, plus immune system, plus liver or lung slices in the most elaborate cases. And we can create a little metastatic event if you have a tumor in the dish, and we can replicate that 96 times to do a screen. Okay? It's not whole animal the way the flies are, and we can't really do the numbers that we can do in flies because the tissue is limited, but it is a great secondary play with the flies. Okay? So we try to set up a pipeline. I'm going to talk about on Wednesday something, yeah. Of course, totally.
The idea of assembloids is mirroring the human, like you've gotten tissues and these different organs from like the hospitals and everything. So you're trying to mirror what you have achieved in the flies in humans.
Yeah, so the question is, are we, so I said we're matching the two, and that's I think what you're referring to. Matching the human assembloids to the fly avatars. In the perfect pipeline, yes. So that we would take, so in a weird way, we construct it in the other direction. I should say this is all new that we're sort of beginning to build now. So we take tissue from the patient, but we also, and when I say we, this is the royal we, okay? We take the sequencing data from that same patient and so on, and we can build a fly avatar from that that then matches the human assembloids. So then we can create a pipeline that, if you turn it around, starts the screen in flies that should match that assembloid. We will get multiple hits in the flies, and as much as I love flies, we're not actually trying to cure fly cancer. So we need a check. And so the assembloids, despite how cool they sound, they're not perfect. They are missing all sorts of things, right? So ideally what you want is something that works in a whole body, fly, and still works in a human system. Yeah? And this is the vision and which we've been working on, okay? Okay, and then the third piece to that, as I'll explain on Wednesday, is something we call a computer, which we're working on with a guy named Lee Cronin, which is digital chemistry. I am not going to explain this here right now, but basically any compound you can imagine, you just type it into a computer and the computer physically makes it and gives you it back. Okay? Like that. So we'll stay tuned on Wednesday. Okay? We'll get into this. And I should say the person who leads that is that guy. Okay? And then I'm going to talk about a fly to bedside clinical trial today to really sort of tie together the various things that you can do in fruit flies. And hopefully it will give you a sense of what's possible. this work? Thank you. I'm old and hard of hearing. You're going to have to, I know you all said yes, but. All right. So as I mentioned, I was actually a developmental biologist. I got in a conversation with a neighbor who studied this thing, medullary thyroid carcinoma. Very long story short, people in my lab ended up basically working with AstraZeneca to develop a drug that has now been approved. And I'm going to explain briefly how this is done and how you use flies to get at this. Especially, this conversation has now come up multiple times. Some of you are doing drug discovery, so let's talk about it. All right. So medullary thyroid carcinoma, thyroid cancer comes in two versions. The not a big deal version where you have a lump, and they're even starting to leave those lumps until it gets to be obstructive and they pop it out. Okay, that's the more common one. But there is a version. There's actually a couple of versions. One of which, the more common one, is medullary thyroid carcinoma. This is a tumor that you will probably die from. All right. So it grows in there. It metastasizes elsewhere in your body. And like I said, it slowly walks you down until it gets you, and you will die from this. And I should say our efforts, if you want to look them up, we were featured on CBS Sunday Morning when TV was a thing back in the day, 2 Esquire magazine articles on our later work that will also describe these things. So if you forget about this, you can just go there. And it's also me doing a humble brag about the media. So medullary thyroid carcinoma is primarily driven for mutations in the RET receptor. So RET is one of those canonical receptor tyrosine kinases that when you turn it on, it turns a whole bunch of stuff downstream, starting with RAS and then going out from there. Okay? And these were the early days. So our first paper we published with AstraZeneca in 2005. Thank you. 2005. Okay? And what we did was, and this is, again, the royal we. So AstraZeneca was the one who really did the chemistry on this. And we basically screened and identified a compound called ZD6474, which now has a name, vandetanib. It also has a commercial name, Proprelsa. And this, for a while, was actually standard of care for thyroid cancer. It has now been superseded by better drugs, selpercatinib, which are more potent RET inhibitors. The punchline here is that it's a RET inhibitor. It probably also works by hitting other things in the pathway as well. It's what you would call a dirty drug, vandetanib. I am the king of dirty drugs. I love them. All right? This goes back to my original conversation where you see target, hit target is a problem because the tumors can get around this and it's very toxic. And so I actually prefer drugs that are what I call network-based drugs, which hit lots of targets. Actually, most of the successful drugs actually work that way, despite what's written on the label. Okay? So when you have 2 RET inhibitors that go into trials and one succeeds and one doesn't, the reason is all the other stuff that the successful one does that they probably don't even know about, but that gently moves the network from here, which is bad, to here, which is good. And this, again, is the brilliance of screening. I don't know how to take a network and hit this target 40%, that target 70%, and so on, to start moving networks around. But the fly will tell you. Just screen, it will show you the drug. In almost every case, when we do that, we end up getting dirty drugs. Okay? Or, sorry, polypharmacology. All right? Now, polypharmacology has some pushback against it, and we had a lot of pushback, which I'm happy to talk about if you wish. But that has really been what we've been most focused on, I would say. Skipping a whole bunch of stuff here. Let me explain. Okay, so the second one, no. The first one, vandetanib, yes, in that the original paper that described it came from my lab. Okay, and I'll show you this. Just give me 2 slides, and if I don't answer it, please come back. And the way we did this very simply was, it's a little bit of a story. We had developed one of the first fly cancer models, which is basically we took human oncogenic RET, so the disease form that causes cancer, stuck it in a fly. I didn't know anything about cancer, so we stuck it, and when I say we, I mean Nancy Novak and then my student, Renee Reed, put it in the fly, expressed it just in the eye, Why did we do that? Because we were studying the eye. We didn't know anything else. When you put it in the eye, it turns out you get eye tumors. Okay? And then at that point, we were approached by somebody named Sam Wells, who represented AstraZeneca, and they said, we have a set of compounds. We don't know what to do with them. We think some of them might be useful. We screened those and said, this is the one you want. Okay? So it was really a team effort. Does that make sense? And so if you feed the fly vandetanib, just mix it in the food, you can see this eye, that should look like this, now looks more normal. This screen was just that. Okay? And that worked. Here's the original. So it was originally ZD6474. This is the original paper. Marcos Vidal was a postdoc in my lab who did the original work. Sam Wells, the guy I mentioned, Anderson Ryan is from AstraZeneca, and that's me. And so it went from AstraZeneca through clinical trials and was approved in 2011 as the first treatment for thyroid cancer. Okay? And honestly, at that point, I was kind of blown away by how well this had worked. And so I was like, screw this. So I moved the lab to New York because that seemed like a good idea. And we flipped from development to drug development. Does that make sense? So very simple, nothing dramatic about it, but it worked beautifully. And again, vandetanib, what does it do? It does inhibit RET poorly, but it inhibits a lot of things. And it was actually quite well tolerated. So you would think that if it's dirty drug, it would be more toxic. This is something I have an argument with all the time because people think very linearly. Oh, you hit one target, it's toxic. You hit two, it's doubly toxic. It's not like that. I hope you understand the concept of a network, all right? Okay. So I'm now going to move to fly to bedside. We're good with this? So that's how you basically screen things. You mix it in with the food, you let them eat it, you set up a quantitative assay. In this case, we just scored the eye get better. Usually now we score where the fly dies of its tumors and then we unkill it. It's a much cleaner screen. You get more quantitative data, whereas phenotypes tend to wander around and then it gets to be a bit of a mess. But that said, this is our basic approach. How do we unkill the flies? Right. So, flies, as you'll learn, they have a life cycle where they're laid as eggs. They can't eat drugs at that point, so you're not curing embryonic cancer. Then they hatch out as larvae, which are basically little mini worm-like adults. At that point, they're eating machines. They're brilliant for feeding them drugs because they eat in an uncontrolled fashion. No matter how nasty the drug tastes, they don't care. They'll just eat it. Okay? So you mix it in with the food. So usually by the time they become adult, they'll die because tumors will start to grow in them the way we build them. And I think you're going to really explain better how practically to do this. But basically, you hook up cancer genes to a promoter that just turns it on the eye. We focus a lot on the gut now, so it just turns it on in the fly gut because we're studying colorectal cancer. You get these tumors that become metastatic and invade the body. And my lab has published a number of papers on the biology of all this. And they'll die of this, okay? Like us, they don't like having tumors taking over their body. So now you feed them the drug, and then you wait till they're either pupa or adults, and you count. I expected 30 flies to come out of here. I only see 3. So I've got a 90% kill rate. Now I feed them the drug, and instead of 3, I see 7 on average come out. Okay, that's not a perfect drug. Pretty good. You take that, maybe modify it, maybe get other ones in that space, feed it to them. Ooh, now I'm up to 15. So we walk it up to as high as we can go. And that's how we do it. We don't obsess to getting it all the way to 30 because we're not trying to cure fly cancer. It's just putting you in the proper space. But that's the idea. Make sense? And let me, while we're on that topic, a little lingo, since you're all going to be drug development people now, I assume. So it's a drug after it's FDA approved. Before it's FDA approved, it's a compound. Okay? You're not screening drugs, you're screening compounds. Unless you happen to have a set of FDA approved drugs, I wouldn't recommend that screen because the patent is usually dead by then. Nobody is going to buy that drug for you and put it into clinical trials. Unless you have $500 million lying around, you're not going to run the clinical trial. We can talk more about that. Okay? But just for the lingo, compounds, means pre-drugs. Okay? Let's talk about fly to bedside. And this too was a little bit of a story which I will not go into because I say I'm going to run out of time. Basically, in interacting with some of these cancer patients from that original thyroid work, I became motivated to see whether we could push fly technology. And when I say I was motivated, I was contacted by a medullary thyroid cancer patient. We had a discussion, and I realized we needed to do more than just spit out drugs that worked for a while, and eventually it fails as well. Both of those will eventually fail, that I showed you. And this was annoying. So long story short, we developed a method. And again, this is the Royal We, it's a team, where in short, this was all done in New York. So we moved to Glasgow from New York about 6 years ago. Patient comes in, we sequence them, we do a number of other analyses on their tumor tissue, and we, based on that, we create a personalized fly avatar for each patient. So an avatar means a representative of you, okay? Some of you have seen the movie Avatars, it's that, right? So, and we developed technology to make not a one hit or a two hit, so altering one or two genes in the fly, but a technology that we've never actually pushed the limits. The most complex patient we've done is 16 hit. That is everything that's a potential cancer driver and more, so-called passengers, things that aren't even supposed to matter, those go in as well. Everything goes in, okay, and it all matters. If the fly is diabetic, we would feed the fly a high sugar diet. Some of you mentioned diabetes. My lab has actually studied diabetes in some depth, okay? And that affects the tumor and affects drug response. If the fly is a Republican, same thing. All affects it. Everything goes in. The Republican, they take their chances. So once we make that fly avatar, so why flies? Okay? I'm often asked, why don't you do this in zebrafish, right? So let's talk reality here. Once we make that fly avatar, this is a clinical trial. These patients are dying by cancer. We don't have a bucket of time. And to do a screen, we developed a fully automated robotics-based screening system, okay? And we grew up for each patient 400,000 flies. And you're going to have Laura speaking here, yeah? So a company spun out of this Vivant, which she'll talk to you in more detail about this. So we grew out 400,000 flies for each patient. So a zebrafish will take, I calculated, 8 years to do that, okay? Let's not talk about 400,000 mice. I don't know how much that would cost, but it would be funny to try, okay? There is no other system you can do this in. That flies are it. So flies, it takes us 6 weeks, all right? That's not perfect, but that's not bad, all right? So 400,000 avatars of you and your cancer. And then we whip out the robotics. We do drug screening, or in this case, it was drug screening because we couldn't treat them with experimental drugs. So what we did instead, because we knew that no single drug would work, that's why if you come to a fruit fly clinical trial, you're in trouble, right? You didn't come to us because things were working. And so what we ended up doing is screening through multiple rounds to create a 2 to 3 drug cocktail personalized to each patient. And that's the way we came up with a new drug for each patient, was to come up with crazy combinations, okay? And then once we came up with the combination, we sat down with the tumor board who said, yeah, this seems safe enough, and we treated the patient, right? Yep. When, somebody had asked about what this exactly looks like, so I just popped this in here. Here is what a fly gut tumor looks like. So here is the fly hindgut, here labeled in green with GFP. This is the muscle wall. And incidentally, you're more than welcome to take pictures, but I will be happy to send this deck to you, okay, as a PDF, just FYI. Muscle wall. This is trachea, carries oxygen like our arteries. And here's a cell that was here that has now pushed through the muscle wall, hopped onto the source of oxygen, and is leaving. This is actually its pathfinding process here. It's heading off in this direction elsewhere in the fly to form a metastatic tumor. Okay, so this is what I'm talking about. This happens to be an easy 4-hit, only 4-hit tumor. Okay, this is like low-level complexity. Okay, so this is what we're talking about here. So here's a patient, failed all standard treatments, came to us, 53-year-old male with advanced disease, was actually in, I think he was actually in hospice or was heading into hospice. We made a 9-hit fly. These are the 9 genes that we modeled in the fly, targeted to the fly gut tumors. Okay, just notice that this is bigger than that. Okay, did the screen, came up with a 2-drug cocktail, a cancer drug, trametinib, and a bone reabsorption drug that women take when they have problems with brittle bones, called zoledronate. I want to point out flies don't have bones, so it's clearly not working on that. Okay, but these two together, either one alone didn't do much. Together, it was magic in the fly, it was magic in the patient as well. Here's an example of scans. Week 0, this is not in the gut, this is a metastasis. It's actually pretty high up here. Pressing in the spinal cord, this man was in serious pain. Okay, this is one of 14 lesions that we followed. And 27 weeks later, you can see it not just went down, but stayed down. He actually went into remission for about a year, went back to work. Quality of life was actually quite good. And then after about a year, he came back, frustratingly, and he did eventually die of it. So this was both a win and a loss. Okay, but it's a step forward. And I have, other stories like this. Sometimes we were successful, sometimes not. I'm going to leave the fuller stories to Laura. I will show a second example on Wednesday that's relevant to what I'll be talking about. But this is the basic approach that we used. And what it told us was, first of all, flies can actually be useful, right? It's no good me running around telling everybody how great flies are. I actually also myself needed to know, can this actually model a tumor to a level that's useful? Okay, and I'm gonna. Am I okay time-wise? I'm gonna do a. I'm okay time-wise. What am I? How much time do I have? 16 minutes. Okay. So let me just say, and this is just me saying for myself. When we worked on developmental biology, it was all about mechanism. Mechanism was king or queen, and you got mechanism. That was the point. But in disease, that's actually not the point. It might be one way station to getting to the point, which is treating it. And I'll give you an example of what I mean. My dad had a heart attack, okay, and he had quadruple bypass surgery in his mid-60s. He lived for another 23 years and died of dementia, not of heart disease. What causes heart disease and what caused his? Not clear. Didn't matter. They just bypassed around it. He got to live. And I always remember that. Okay. Side note, Dad was a great guy, and he had a great extra 23 years, which I'm grateful for. But you see my point. Okay. So for me, I never try to forget the point of this, which is treatment. If I don't understand why that works, that drug combination, but it actually gave that person time, that's a win. If I understand perfectly his tumor and I have nothing to offer him for it, that's not a win. Okay, that's just me. Okay, so let's move forward. So as I mentioned, 84 European avatar lines just for colorectal cancer. We have avatar lines for rhesopathies. We're starting to build some for other tumor types. We got a lot. I'll show you on Wednesday, adenoid cystic carcinoma, a whole bunch of avatar lines. We're all about avatar lines, okay? And I can recommend them. They're really lovely. And with Amos, we've had the privilege to begin to build a Nigerian set as well, so that we can ask the question, treatment-wise, are they the same, right? And I can tell you, I know you'll be surprised, that West African patients are underrepresented in the databases. Fortunately, that is changing, okay? And that's how we were able to build these avatars. We didn't do the sequencing. So Peter Kingham and Alates have done some sequencing of Nigerian patients. So this is the good news. we've begun to build avatars with them, and we're actually we put in a large grant with Amos to begin to really expand that quite a bit, do spatial omics and all this stuff. Okay? So work in progress. Now I'm showing Amos's work and his team's work. So for example, RAS APCP53, a 3-hit tumor, plus additional mutations in Nigerian patient number 3. And you can see already, if you're looking at, this work? Yeah. I don't know if you can see that, but it doesn't matter. Basically, if you're looking at things like oxidation, gut metabolic rate, and so on, when you treat these flies with standard of care drug called regorafenib, which is the only targeted drug approved for these tumors, and it works so poorly, it's kind of a running joke. You can see why here. Basically, these flies are cranking up their response to it to get it out. So they're cranking up their mitochondria and so on. And this is probably why this drug is having trouble doing anything. So these are the sorts of things that we are hoping to follow up on and learn more about. So I'm going to finish. And I, of course, assume that many of you, maybe all of you, will help us in this effort and build more fly avatars. So I'm going to finish by asking a simple question. And that is, if we take our fly avatars, if we take our human assembloids, not just us, but others, and we pull the major driver of it, KRAS or RAS. RAS is generally known in the cancer field as the Death Star of cancer, okay? Because until recently, we had nothing to drug it. It was considered to be undruggable. It didn't really have docking sites. That is beginning to change now, which is great. The first ones that are getting approved that are targeting it give you up to 5 weeks of extra life, so there's that, but it's a start, okay? But that's actually very confusing, that these things don't work. So trametinib is a super potent, what's called MEK inhibitor. It's 2 enzymes down from RAS, completely shuts MEK down. And I can tell you genetically, if we remove RAS or remove MEK from these tumors, they stop. If you drug them with a super potent drug that hits the same target that your genetics did, they don't. Why? Why is that? Okay? So now we're starting to crystallize what we've been talking about down to an addressable question. We give the drug, it's supposed to work. We know if we get rid of the target that works, but the drug is somehow not working. What's up? Okay? So I'm not going to show you data. I promise not to show you much data. I'm not going to do that. But let me tell you, if we have a one-hit fly, just activate RAS itself. The drug works. Actually, all those drugs work on a RAS, one-hit RAS alone. So they can hit the target, all right? If we move to a 3-hit model, this is the most common 3 mutations that you'll see in colorectal cancer, RAS, APC, P53, and you give this or any other RAS pathway drug, won't work. Okay, now we're learning something. You add some mutations, something changes. What is that? Yeah, so now we're starting to walk it down. So what we did, this is Boji Kong, former postdoc in my lab. He's now a professor over in Hokkaido. Or in Harbin, he ran a mass spec. I am mercifully not going to go through the details of this. And he just compared RAS alone to the 3-hit model. What's different? And basically, what turned out to be different was a pathway called glucuronidation. There's Boji. You can tell when this picture was taken. So what he found was a pathway called glucuronidation. Who here knows what glucuronidation is? Do you? I had no idea. You too? Respect. I had no idea what it was. But this is the beauty of doing screens and flies, of course. You come up with this stuff, and then you got to get online and figure out. Glucuronidation is a primary way of modifying toxins and getting them out of the cell and kicking them over to the liver. Exactly. Okay? So you stick a UDP glucuronate on your toxin, stick it on there, and that targets it to get it out of the cell and off to the liver where it gets degraded. Toxins like a cancer drug, for example. Most targeted therapies are glucuronidated and dumped. And what Boji showed was, so that's shown here. Drug comes in, gets glucuronidated, kicked back out. Okay, this is my detailed map of the path. Glucuronidation is so complicated, it's crazy. So this is the core of the glucuronidation pathway. Here is that drug I was telling you, trametinib, that should work. It works on the 1 hit, won't work on the 3 hit. It's glucuronidated on 2 sites, pretty typical. And what he found in his mass spec is that 3 of the key enzymes, when you add a knockout of APC to RAS, when you knock out APC, you drive up a pathway called Wnt. RAS plus Wnt drives those enzymes up. Glucuronidation gets pushed up. And now you can feed the fly anything you want. It's just going to get glucuronidated and thrown back out. And that's why it's not just resistant to this targeted therapy. It's resistant to everything. Because anything that goes in there is a toxin. The cell's like, well, this isn't going to work. And I've now cranked this pathway up. Bring it. Okay? And actually, that works. So if we knock down this is the royal we, this is OG. You knock down any of these enzymes, like this SGAL, just bring it back to normal, where before you added that APC mutation. And now fly survival goes way up. In other words, the drug is starting to work again. Okay? So I hope that's clear how we used flies, whole body, to get a basic question of like, why is it that not only does this drug not work, it seems like nothing works on these things. And why is specifically, so very commonly, you'll get a RAS pathway inhibitor, works great in lung cancer. We see this, I see this in the newspaper all the time. Works great in lung cancer, works great in melanoma, doesn't work in colorectal, to the point where we just assume it won't work in colorectal. And it never does, and that's why. Because they don't just activate RAS, they also activate Wnt. That's a magic pairing. Boji worked out the detailed pathways in flies, which I will not show you, out of mercy. But he worked out the detailed pathways of how RAS plus APC activates glucuronidation. Okay? What I won't discuss, because I know I'd be running out of time, this is one pathway. A student in the lab, Bjork Aston, I'm not going to go through her work in detail. What she showed is that somebody had asked about blood cancers. What she showed is that fly macrophages, I'll use that term loosely. I'm sorry, this was actually started not in flies, it was validated in flies, but it was started in mouse cell lines. But what she showed is that macrophages actually send a second signal to drive drug resistance. It secretes 2 factors, GDF15 and PDGFBB. And we don't know if they are then activating glucuronidation, but it would make sense because they end up intersecting at the same pathway as the thing I showed you. Okay? So everything, I think, is getting together to crank up this detox pathway. And then it's like, good luck, feel free. So you understand that no matter how much research you do on a really great new drug, if you don't solve this problem, it won't matter. And if you do solve this problem, I think many of these drugs that failed actually will start to work. And we'll find out. Doesn't matter what I think, we will find out. We're heading in that direction. For example, we're working to make a non-glucuronidatable version of trametinib, okay, with my buddy Arvind Dar. Okay, and is this reality? And this is where I would really urge you to open. We're talking about this morning the difference between learning vertically, one thing, and learning horizontally, many things. I think as the new age of biomedicine, you need to be more horizontal. Okay? And so, for example, we asked the question, all that stuff I showed you, which is a series of enzymes that get driven up, does that happen in people? And it turns out, if you just pair human KRAS with human loss of APC, just like I showed you in the flies, if you pair it versus not having them paired together, mutations, you live less long and you respond poorly to drugs. In fact, you have double the chance of dying at any particular moment if you just have that index. So we think this is actually what's happening with people. And similarly, I very quickly went through this bit with macrophages secreting these 2 factors. Same thing. When you get that fingerprint that shows that they're starting to secrete those, then you respond to drugs poorly and you live less long. Okay? So that's a good check for us. We're not just curing flies now. Now we have something that at least correlates with poor survival. All right. So let me wrap this up. what's next for these projects? What's next is spatial omics. And flies are magic for spatial omics. My talk on Wednesday, I'm going to talk about pairing big data with flies and why they are magic. The punchline of that will be that you can actually screen like the entire fly kinome in weeks. You can take your favorite 500 genes that came out of your big omics project and check all of them in flies. That's something you can't do in other systems. And so instead of taking your big data and going, yeah, I study that, that looks important, let's focus on that, which is like, what was the point of doing the big omics, the big data search in the beginning if you're then going to just cherry pick what you think is interesting? Why not take the set, check it functionally, and learn something new? And flies are magic for this, okay? And that's what we're doing. We have a huge, and again, this is Royal, we have a huge project. It's part of a cancer center that I put together called the McNabb Center for Cancer Innovation. And part of that is bringing these big data, spatial omics online and bringing it to things like flies and so on, and human assembloids, okay? So, and of course, starting companies, happy to talk about those. I think Laura's going to talk about a company that's spun out of the clinical trial. We also have a company for rare diseases. You're looking at the chief operating officer. And my final comments on this talk. So again, old enough to remember this, let's talk about the computer age. 1950s, 1960s, they were working out information theory. 70s, they were optimizing the silicon chip. Basically, in academia, they were working out the basic fundamentals of putting together a useful computer, okay? And then at some point, really starting in the 90s, governments said, that's about how much we're going to pay for that. Good luck. And in the 80s and 90s, the Apple, Microsoft, all of those groups opened their garages and started to build the next world, right? And they built, my phone's over here, my big dramatic reveal. They built this, okay? Because they cared about it and they thought bringing information to the world mattered. We're now in the biomedical age. A lot of the basics have been worked out, not all, never all. But a lot of the basics have been worked out. And I recommend continue to work through those. It's a ton of fun. I love being an academic. But we're now moving into that next phase. And my colleagues hate it because governments are like, this is about how much we're going to pay for that. Good luck. All right. So we like our grants and like to bitch when we don't get them. Okay. But that's not your world. Your world is that next phase. So some of you will go into academia, but many of you will go into industry. Many of you will go into, here's my clicker. Okay. So basic research, biotech, but also financial aspects of it. That is now a huge thing. And actually, I had a program at Mount Sinai in New York where I had a pipeline from getting your PhD there to going straight into Wall Street. Okay. Teachers, public and social media. As we know, the new world of medicine scares a lot of people and has somehow become political. We're going to need you all to explain to the public what we're talking about. They still think what we're doing is a good idea, but that's not going to be for forever, okay? There are people that doesn't bring advantage to that are going to erode that trust unless we speak up. And in terms of the fly, we're going to spend this week talking about that. I hope I've convinced you, you can do elite science without elite funding and without elite infrastructure. You had mentioned that we need to build more, and you're exactly right. But the other thing I got from that story, from yours and from Dr. Fala, was that you had the problem and you solved it. You learned how to do the Western blot. That's actually what I got from it. So we have fallen short in providing those. That is going to be the case. Until that's fixed, some of them you're going to have to solve yourself. And so I actually thought that was excellent that you got through that. It's a great model for teaching. And actually, we've had in East Harlem, we had a whole thing where we actually brought students into Sinai into the teaching. Hopefully, we're going to be doing the same in Glasgow with my cancer center. And I mentioned, finally, it's a great pair with other models, and you can find collaborators locally and internationally to collaborate. And people, other researchers, love fly people. We can generate data like no tomorrow. And then we give them the data, and they're like, great, I'll just test it in my mouse, and let's see how that goes. So I'm telling you, we're like their favorite people. We're like data generators. So all of that. And last slide. So what I talked about today, flies represent a chance to do whole body biology. Don't let anybody tell you that other ways of going about it are better. That's just because that's what they work on. Whole body matters. I showed you an example with macrophages. I don't know anything about macrophages. So a lot of the things you're going to work on impact the whole body. So I'm going to talk about diabetes. That has a huge impact on cancer. Okay? I gave an example of vandetanib, so I'm not just making this up. Fly to bedside. And we'll talk on Wednesday about how big data, flies are a perfect match for big data to allow you to actually extract the useful thing of big data and not just, look, I did big data, okay? And then cherry pick it. As I mentioned to Dr. Fowler, I started 4 companies. There's 3 of them. You're going to hear from a founder of one of them, Vivan. Flies are lovely for starting companies for all the reasons that we talked about. And you have many options career-wise. And I think we're going to get, I don't want to get into this in detail, but we hope to bring to you some like solid points on what happens next as you walk out of this week, where you can go and where you can take these things. And you can genuinely build a career from flies, but like I said about horizontal, it can't be everything. But it can, if you're the best at flies, you can be a key cog and you can do really kick-ass science at a high level. Okay. That's why, despite the fact that I don't know how many people are left doing flies in the lab, but we'll always do flies because they're the feeder to everything else that we do. And I think that's it. So thanks. Thanks for your questions.
Wow, amazing. I'm sure our notes are fuller than it was in the morning. So do we have comments, questions from online participants? And then we'll take 1 or 2 from the house because we are behind schedule. Yeah, question.
Yes, sir.
I wanted to ask, what's the difference between creating these personalized fly avatars and the assembloids? Like, what's the major difference?
Okay. So the question is, what's the major differences between the fly avatars and the human assembloids? And the difference is, so what's very quickly, the pluses and minuses of each relative to each other. So fly avatars, the advantage is their whole body. This really matters. I just gave a speech on that. Flies have livers, they have lung-ish, they have guts, they have kidneys, all of this. They get diabetes, so they have the basics of us. Most of the things that drive disease are fundamental to the body and will be shared across species, okay? And the other advantage to the fly avatar is it has many or most of the drivers of cancer, okay? So we can make a 10, 15 hit fly. So that's the fly avatar. The disadvantage of flies is that the details are different, and in drugs, details matter, okay? You can't have a drug that kind of works. It's got to really work, right? And that takes us to the assembloids. Assembloids are better than just culturing cells or doing organoids in that you're adding some back. You're getting some immune system back, something for the cells to interact with tissue-wise, liver or lung slices, things like that. The disadvantage is there's a long set of other things that they're missing. And we've actually, in the past, when it's a complicated story, which I won't get into the details, but we looked at a compound that hit 6 things, and it looked amazing preclinically. So we used the fly to map out each of the 6 activities, where in the fly each of the 6 was required. 3 were required in the tumor, 1 was required in the local microenvironment, I think. 1 was required in the muscle, 1 target was required in the liver. The muscle one, for example, controlled metabolism, right? Which kind of makes sense in retrospect. If you get rid of any of those genetically, the drug didn't look like much, or the compound didn't look like much of anything. You would not pick that up in a human assembloid. There's no muscle in there, all right? But you would pick that up in a fly. Yeah? So that's the ups and downs of each of them. So, And I do worry when we test what we get in flies and the human assembloids that we're going to kick out things. But I don't know. I mean, they're not going to let us go from test it directly in humans. So yeah. So in a perfect world, so when we have the assembloid, we can get the sequence. We have access to that, so we can make the fly avatar. And the idea then is they should work hand in glove, right? You can argue that you don't want things to be perfectly matched because then you're curing one patient. That's why we'll have many examples of that. But if the assembloid is very different from the fly, then we'll end up getting rid of everything. So there's a balance. Yeah.
Any other question?
Thank you, Professor Cagan, for your presentation. One, I have two contributions or question I want to add. One of the drug you discovered or you discovered in your lab, you said it's an glucuronidation which limits its activity. So I'm talking about if you do drug optimization, it can actually reduce the process of glucuronidation, and that may make the drug work better.
Yes.
Then two, for example, in Nigeria, or let me say in
I'm sorry to
Africa.
interrupt you, but can we stick on that for a second?
Okay.
Do you want to answer drug optimization and what you're doing for it? Do you think you want the?
So we've got a system in the lab that we call chemical evolution. So these allow you to play with the atoms of this compound, and you can modify whatever you want to modify.
Yeah.
Oh, good. Oh, I see. Hmm. Yeah, good. Yeah.
Differentiate it based on the function of what the central compound still remains.
Yeah, that's what we do.
That's what you do.
I use the neural network.
Oh, I see.
Whereas you the compound will have example recent one published is not go ahead testing it in any model. I just build using silico to detect its therapeutic action, then just throw it out because we don't have the facilities to test in the wet lab. So I'm not an biochemist, I'm not a chemist. So another things I want to ask is most of the compounds you got, I think they are known compounds. So I'm thinking you working on a novel compounds.
We do. That's
That is
what we do.
Then secondly, do you have you look at getting a natural compound? For example, Nigeria,
Now
we are
you stepped
blessed.
in it.
We are blessed with a lot of
I'm
natural
aware of
compounds, flavonoids, terpenoids. So have you tried looking at the central ring?
How much time do I have? 2 minutes. I'm going to give you the 2 minute because this is going to be the same. So we yeah, you said that the magic words, and so we mostly build novel compounds. What we don't do is we don't build them to hit a target. So we don't do any predictions on that. We only explore the chemical space. We end up with polypharmacology. We can talk more about that, but that's what we care about, and for all the reasons I described. Let's talk about. Let me do the 30-second. Okay, and why natural compounds is not what they're promising you, with apologies. You check me on this. Go ask a pharma company. They have no interest in natural compounds at all. And the reason is the chemistry is too complicated. Okay? You cannot extract and run a clinical trial, so let's not even have that discussion. You need to make it. It needs to be at 100% purity, done at GMP level so it's safe for a person, and then run a trial, and it can't be expensive. You might say, but you're going to make $1 billion, so it's going to be worth it. The current success rate, which I will go through on Wednesday, of cancer clinical trials is 5%, right? You have a 5% chance of being, I don't know why I'm talking to you. You have a 5% chance of it succeeding, and that's after it's passed everything preclinically, right? They are not going to spend, as somebody said, last time we did this with Taxol, it took us 25 years to figure out the chemistry of Taxol. We will never do that again. Now, there are new ways of doing chemistry. I'm not an evangelist on this, but make sure you have an answer to that question when you approach pharma, who will run the clinical trial. Again, I don't know why I'm looking at you. Well, that when you say, I have a natural compound for you and it's going to cure cancer, make sure you answer that question. It was a little, okay. Yeah.
Asked this question last week during the neuro track, but I think it would be best to ask it in this track. So, in drug discovery, during the in silico phase or in silico studies, usually what we look at is your drug not being able to cross the BBB, the blood-brain barrier. So in flies, I don't know if you could model this, but let's assume we've done everything in flies. We are trying to advance it to the human trials, that's the clinical trial phase. How do we confirm this drug that you are trying to develop wouldn't cross the BBB in humans, assuming you are trying to treat a certain cancer?
Would not.
Wouldn't.
Would not.
Yes.
Okay. So the question is, can you use flies to determine if a compound or a drug is going to cross the blood-brain barrier? The answer is sort of yes and no. Flies do have a blood-brain barrier that Ulrika, at Rockefeller showed many years ago. So they do have a blood-brain barrier. It is somewhat like ours, but not perfectly like ours. And I would certainly not go to a drug company with only fly data showing that it doesn't cross the blood-brain barrier in flies, because there is some differences, in part because they've got something called trachea that goes in, and it's possible that the drug will track its way through. But they do have one. It shows some aspects. It will exclude things that are commonly excluded in us as well. So the answer to your question is, I wouldn't go to the next stage with just fly data, but they're a decent initial call. You will, of course, need a way to visualize the drug. You can talk to this guy who has ways of visualizing direct drug target physical interactions. And I don't know if I'll talk about it. So that's really the answer. So kind of yes, but kind of, yeah, you'll need something. And there are pretty good in vitro blood-brain barrier tests that you can do that I would suggest you show up with those more than just flies. Okay?
Okay. Before I ask the question, I need a yes or no answer, please. You talked about talking about how you get the flies to take the drugs. Do you you said you put it in their diet, right?
Important thing is that they
In their diet. In their diet. Okay. The drug.
in their food.
In their
It goes in the food. Yeah.
Okay.
Diet.
Yes.
I thought you said that.
How do you do you is it the concentration doesn't matter? What if they do not finish the food? What if they do not finish the food? The diet in which you put the drug. What if they do not finish it? Does the concentration matter? And in what form do you give them the?
What do you think, Hamid?
Yes. Concentration matters. Short answer to that. And they don't need to finish the food. So basically, you're feeding on food. We want them to have something in your system that can work. So we play with the concentration, like tenfold changes, and then we can see what happens. And a lot of them will work. Yeah.
And also, when you work, you work in solvents, appropriate solvent, then you mix with the food. Then, when you are mixing it with the food, we refer to it as the concentration. The one you give rats or human that will take the whole thing is the dose. But with respect with the file is with concentration. That concentration has to be the final concentration, putting into account the volume or the amount of the diet that you are using.
Yeah.
We'll get into that in the course of the
we estimate that the concentration in the food is 200-fold what ends up being the concentration in the fly blood with human, roughly speaking. Yeah.
Round of applause.